And last speaker for the day is Matthew Buddy. He's going to talk about biophysical interpretation of the diffusion contrast. Thank you very much.
Thank you, Flavio, for the invitation. It's nice to follow such an esteemed group here. A lot was covered today, and so I want to try and give some context into when you have a diffusion measurement and you see some changes, how do you interpret that? What's going on at the really microscopic level? So again, here's my obligatory nice picture slide. I think this has been well covered, but it is truly an amazing process that really what we're looking at is a very fundamental physical process, just water molecules moving around. And from that, we're able to generate these really intricate and really astounding images of the human brain in vivo. I mean, no other technique is really capable of this. But as you heard along the way, there are many steps involved in this process, and so we have to be careful of moving from this physical process to our representative data. And so here's some perspective. These are our 3D electron microscopy data sets from animals, from fixed tissue. So if you look at the white matter here of an animal in the external capsule, you can really appreciate just the extraordinary amount of detail that's going on here. So you can see a glial cell at the bottom, and just a number of white matter tracks interspersed with one another going many different ways. It's quite revealing, and so again, just stepping back and trying to capture all of this, we're not really able to with current technologies. And if you think that's a problem, look at the cortical gray matter here. It's just, again, pretty astounding at the amount of complexity here. So in any diffusion technique, whether it be DTI or some other complex model, we're really getting a composite average in the voxel of what's going on, even if we're looking at multiple fibers or multiple different tissues. So I'm going to start with the take-home message. Again, you've heard this over and over today, but we're inferring structure from our measurements. And so really this takes, as you heard, a process everywhere from the measurement to the model to the mathematical quantity or physical quantity that we're trying to represent, all the way through validation. And it really takes converging evidence for us to really understand what's going on at the microscopic level. So just a brief outline here. First, I'll talk about some histological studies that I've been involved with and others, looking at orientation or connectivity. So how does our tracks at the local level match what we see histologically? Second, moving on to what we've heard about a little bit, looking at the normal microstructure, tissue density and diameter, or track coherence and complexity. And then followed up where things get really crazy is abnormal microstructure. So when we're trying to investigate some pathology, whether it be from injury or disease or other modulation, it's really where things get much more complex. So to start out, how well does our DWI, or diffusion weighted imaging, our fiber orientation at the local level match our true fiber orientation? So some of the early studies that have been done to address this is Ligard et al., where they manually traced on some stained tissues from an animal model the individual tracks that they could pick out here. Again, a very tedious process, but they did a really nice job of looking at two different regions here, one in the corpus callosum at the top, and the second in the brainstem, where they see some crossing fibers. And what they show, it's no surprise that if we have a single coherent fiber population in the corpus callosum, DTI and Q-space imaging do both a really good job of showing the fiber dispersion and the orientation there. The problem is, as you've heard before, DTI does not do a good job at the bottom here, where you have two fibers that are intersecting. However, these advanced models, in this case QSI, does a really good job of localizing those two different tracts. More recent work, Jesperson et al., along with Chris Kroenke, have looked at DTI and compared that to, in the developing cortex, using a Golgi stain, they were able to identify, so Golgi stains only stain a selected number of the neurons within the tissue, so they were able to quantitatively take those stained neurons and through some algorithms be able to extract those from the histological images. And then the right, they show their histological ODF on the Schmidt plot compared to their DTI. And in general, they found pretty good agreement, both in the orientation and the dispersion here. Another example, Adam Anderson and his colleagues looked at using image processing techniques, in this case a filter matching, so you can see the small filter on the far right top that they used at every point in the tissue to match that, presuming that replicated one fiber. And what they'd show, again, is no surprise that in a region with a single fiber population, things align quite nicely. However, at the bottom there, again, when you have two intersecting fiber bundles, that DTI does not do a very good job. And they show that quantitatively, their measured angle differences generally correspond, fall to zero, but there is certainly some disparity between the DTI measurements and the myelin staining. Another technique that I'll just mention here is polarized light imaging, where one can use polarized light, myelin, as a birefringent response, and you can use that by polarizing the light at different angles. One can get a map, in this case a whole brain map of the orientation angle here at all points in the brain. It also does 3D. Although in the cortex here, where you see there's no myelin staining or relatively little, it errs on the side of the tissue being oriented through planes. So there's some limitations about needing myelin staining for this. One particular technique that I've used is taking histological images. In this case, they were stained with just a simple lipophilic dye, so it stains all of the membranes. It's not specific for any one protein or anything. And using a image processing technique called structure tensor analysis. And so I won't go into the details, but it's simply an image processing technique where one can derive from the image. It looks at the edges in the image and can derive a measure of anisotropy in the image. And so in this case, applied to a rat brain, this is purely a histological data set. On the left here, you can see the orientation color-coded here, as well as it's scaled by anisotropy. So you can see just the amount of tremendous detail that you can get from this. And this technique, one advantage is it can also be applied on the same resolution that you acquire the images. So on the right here, this is actually one of the small regions that was acquired, and you can appreciate just how interdigitated and how the crossing fibers are reflected here, both in the cortex. You can see multiple different fiber angles, as well as in the white matter, you see some dispersion within each of these little voxels that I've represented here. So what's really fun is I also then applied this to a set of human brain sections. So this is from the Brain Observatory. Jacopo Anis at UCSD in postmortem sections was scanning these 2D sections at very high resolution here. So what you see here is a myelin stain. This is actually inverted, but you don't see too much structure within the white matter itself. When we apply the structure tensor, and again the resolution here is around 4 micron data, so what I'm representing here is really just a zoomed out view. But you can appreciate the in-plane anisotropy that we're measuring here, and so one thing that immediately sticks out, as one would expect, the corpus callosum has a really high anisotropy. And you can appreciate even some of these other smaller fiber bundles that are in here. When this is color-coded by direction, a lot of these different features really jump out at you. And so I'll step through some of the examples here. One of which is the region here where we have multiple intersecting fibers, and you can see how in the color maps here you can really start to appreciate just at the microscopic detail that one sees here. And at the actual acquired image resolution, if we look at these are again color-coded by local orientation, you can just see how interspersed and interdigitated these different fiber bundles are. So it does speak to one element that even if we're using DTI, image resolution alone will never get us down to the level of individual fiber bundles. We need some model that captures different fiber orientations within a single voxel. Here again, even looking at some of the deeper brain structures, the cerebral peduncles at the bottom, as well as the internal capsule here, you can appreciate just the amount of structure, even though we consider this a normal coherent pathway. There's a really lot of microscopic detail here that we can piece out. So again, it speaks to that even if we think we're looking at a single fiber bundle, sometimes there's a lot more structure going on that we don't appreciate. Looking at the cerebral cortex here, a few things jump out at us as well, and that is that fibers that enter a gyri in the white matter typically take very sharp angles into the banks of the sulci. And even in a portion at the deepest part of the sulci, you can see some fibers that are horizontally oriented and some are radially oriented relative to the surface of the cortex. And so if we color code this again, this kind of highlights in more detail anything along that white matter, or excuse me, the dotted line there. As it enters the cortex, it takes a very sharp turn. So that's one thing to consider as well. Here's a region in the visual cortex. Again, this was talked about briefly in the previous talk, but using this technique, we can reconstruct different layers of the cortex and get an appreciation for which layers are more radially oriented. In this case, we're at layer 4 in the cortex. You can see is composed of both radial and tangential fibers. So even looking at the medial temporal cortex here, one can appreciate fibers emanating here from the hippocampus, in particular the subiculum. This small structure is the perforant path that is one of the first pathways implicated in Alzheimer's. So we can even pick up on something like this. And there are several diffusion studies that have purported to show and be able to detect this pathway in vivo or ex vivo. So it might be another way to validate some of those studies. So those are nice pictures, and that's all well and good. So how do we use this to really validate some of our measurements here? And here's a study in collaboration with Idemar Ronen. What he was doing is measuring NAA metabolite, it's neuronal specific, in the corpus callosum in vivo in humans. And so he was doing diffusion-weighted measurements in the corpus callosum. And you can see, obviously, in this region, we have macroscopic curvature, and as well as a microscopic curvature or dispersion. So in this case, the bottom here was kind of his algorithm that when he fit his NAA measurements to the macroscopic curvature in A here, the data didn't really fit with the model very well. However, when he also included a microscopic dispersion component, it fit the data much better. So then we were able to take, these are separate post-mortem sections, but in any case, we're also able to derive a measure of dispersion from the corpus callosum. And so by fitting each small region here to a voxel, we can fit it just to a Gaussian distribution. We can estimate the dispersion from within small little regions here. And so in this case, we have a map actually of local dispersion within the corpus callosum. And so what we determined independently is that his diffusion measurements estimated about an 18% spread in dispersion from the diffusion measurements, and that agreed quite well with our histological measurements. So this is one way of taking histological data and trying to validate some of these different models. Another example using a similar technique from my work is that in the animal model of traumatic brain injury, where the brain is actually directly impacted, we consistently observed an increase in anisotropy in the lesions surrounding the cortex. And so in the literature, many people had argued that the increase in FA was due to axonal sprouting. I think in speaking with any pathologist, one response obviously is a tremendous astroglial response after a large lesion like this. So the question was, did this relate to axonal sprouting or was this glial scarring? And in our case, we applied a kind of a unique tractography method to both our DTI data, which we limited to 2D tractography here on the bottom left, and then we also applied the same technique to a stain for astrocytes in the middle, and then neurofilaments for axons on the right. And one can see a nicer correspondence between the astrocytic tracks and compared to the 2D tractography. And so we went on to do a full analysis here, and really we saw increased FA because of the locally organized astrocytes in this injury model. So again, now moving on to some studies of orientation, we're looking at normal microstructure such as cell diameters, tissue or neurite density, and tissue complexity. So the question is, how well do these parameters reflect their histological equivalence? And just as a summary here, I think if we step back, any of these models that start with a biological foundation are much easier to relate to the actual histology. So even DTI is a mathematical expression of the diffusion-weighted signal. So FA can mean any number of different things: dispersion, or density, or local orientation. But if we start to pull what we know from the biology, many of these models use a model of cylindrical axons, for example. And so they all have their limitations, but if we start from a biologically inspired model, it's not to say they don't include a lot of math, they do. But we start to get at a better estimate of what we can measure from histology. And so to think about this, again, at a large scale, really in the tissue we may have several different compartments. One is an anisotropic compartment, and we could have one or more compartments there. We also might have some cerebrospinal fluid contamination. At the far end of the scale, we might have some restrictions from cells, and certainly we have a contribution from the extraneurite or extracellular composition. So again, these are idealized substrates, but they really provide a much more solid foundation, I think, on which to model the signal in relation to what we know about the nervous system tissue. The problem is, if we include all these in a model, we have anywhere between 9 up to 17, maybe 20, depending upon what we include in the model and how many fibers we expect to have. So I think every model, it's certainly worth pointing out that every model has certain assumptions that they make or constraints that they impose on the model to reduce this problem to a more tractable problem. It's virtually impossible to get a good fit to some data that has maybe 15 or 20 different parameters. So one example of that, Jesper et al., as we've heard, model neurites as impermeable cylinders. And in this case, I won't go into all the details, but what they showed is really nice correspondence between their diffusion estimated neurite density and that estimated from histology, including electron microscopy. So again, starting with a density model, one knows what to evaluate going into the histology. Another example where a group has estimated the axonal diameter, and again from microscopy they can physically easily just go in and measure that same parameter, the diameter, from electron microscopy images, and they showed good correspondence here between their DWI and the microscopy. More recent example is using both diffusion and a technique called magnetization transfer to estimate the G ratio. So the G ratio is is essentially the thickness of the myelin here. And so, again, it takes a complex problem and reduces it to a tractable problem in terms of the histological verification. Now, there are certainly caveats always to any different model, but I think this is a nice advance for us to move forward and really go after certain physical parameters here. Another example from Victor Song and his colleagues is to fit basis functions to the DWI signal, and from that they were able to show that their DBSI is better at resolving narrow fiber crossings than DSI. So the last bit of my talk here, and I think I'm going a little fast, but again, we're inferring structure from measuring the MR signal. I think that bears repeating. The problem is, in pathology, we have a lot of different things going on. And so my advice to you is that if you're doing the study, you are the experts on what you expect to happen, right? So you write a grant and you have some hypotheses about if you see DTI changes or DWI changes, you can interpret those. So there is no single answer to any of this. And so I'll give some examples of where things are abnormal, and we've gone on to look at the histology. But schizophrenia, for example, may be very different than a traumatic brain injury. So I think that the investigator knowing kind of what they're going after is really important here. So two questions that one could ask is, how well does DWI reflect the underlying pathology? Or I've observed some DWI changes in my data set. How do I interpret these? And for a long time, the DTI model in injured white matter, so if we're talking about coherent fiber bundles, is that obviously in normal myelinated axons, we have a high anisotropy, which means the diffusion parallel to the fibers is much greater than that perpendicular to the fibers. And so a number of studies have demonstrated that if there is damage to the myelin, that leads to an increase in diffusion perpendicular to that. On the other hand, if there's axonal injury, that leads to a decrease in the diffusion parallel to the fibers. So Victor Song, again, and colleagues showed that in a model of dysmyelination, that they saw increased radial diffusivity here without a change in anisotropy. In a separate model, they showed that diffusion parallel to an optic nerve injury changed early on in injury, about 3 days after injury, whereas demyelination, which is a much longer process, only showed a change in radial diffusivity at much later time points. The Hopkins group here, Zhang et al., showed in a cleaner model of spinal cord exotomy, where they did this ex vivo, so one can see at 3 days, there's a dramatic reduction in the parallel diffusivity here. And they did a nice job of correlating that with their histological measurements. So the SMI 31 and 32 stain for normal and abnormal axons, and the Luxel fast blue on the far right stands for myelin. And so they showed a nice correlation between the parallel diffusivity and their axonal counts, as well as radial diffusivity and their myelin stain. Another recent study that I like very much is one that is a Cooper zone model. So Cooper zone is an inflammatory demyelinating animal model. And what they did is using electron microscopy, quantify all of these different parameters from the histology, including myelinated fraction, the myelin sheath fraction, the non-myelinated fraction, as well as the extracellular fraction. And just simply ask the question, which of the MRI measurements here was most correlated to these indices. And so this kind of highlights the problem that their strongest MRI correlates are shown here, but there were many strong pairwise correlations between the MRI and histology, and also between the MRI and MRI, as well as the histology and histology. So there's many different factors going into play here, although with studies like this, we can start to address which one might be most sensitive to certain pathologies. Here's an example again from diffusion basis spectrum imaging. Again, it's a Cooper zone model where they showed in the top here, they took a control, I believe this was the optic nerve, and showed that the percent of cell fractions estimated from DBSI correlated quite nicely with their estimates of cell numbers from histology. Similarly, their fiber fraction correlated quite well with how many axons were in this tissue. And so in the bottom here, I believe this is, again, a Cooper zone model, but they showed that DBSI was able to show a change in radial diffusivity, whereas DTI was not. And at the bottom here, showing DBSI was more sensitive to changes in axonal injury, where DTI was not. So some of my work is involved in looking at what changes we see in other models. In this case, we're looking at acute cerebral ischemia. So this is one of the cases where well over 20 years ago, almost 25 now, Mike Mosley and his colleagues showed that diffusion is uniquely sensitive to changes in diffusion at early time points after an animal model where we don't see any changes in T2. And so for these 20, 25 years, it's consistently been up for debate. What's the nature of these changes? And so, nonetheless, it has a very clear diagnostic and a clinical utility. So I think one of the caveats to this is that even if we don't know precisely what's going on, if this helps us make a clinical decision, as this has, it's still extremely valuable. So both sides of those coin are important. And so I like to go back to the other fields. In this case, Tim Murphy et al. In his group in Vancouver. We're doing this in an animal model of ischemia. So they have an in vivo 2-photon microscopy of the dendrites in the rat cortex. And what they saw is that 4 minutes after an injury, they saw this very characteristic beating of almost all the dendrites within this field. And importantly, if they reperfused the animal, this went back to normal. So my question here was, is this neurite beating, is this at the root of the cause of the change in cerebral ischemia? And so for that, this was modeled as axons undergoing this beating process. And basically what we showed here was that a diffusion parallel to the axonal fiber decreased dramatically, which led to a decrease in mean diffusivity, although the diffusion perpendicular to the fiber didn't change much. And so in collaboration with Christian Bellou and Corey Barron at Alberta, they were doing measurements in humans comparing the pulse gradient spin echo, so with long diffusion times, versus the oscillating gradient spin echo, which has, you can see here in their specific example, a much shorter diffusion time of 4 milliseconds or so. So there were two possible outcomes that we sort of considered here. One is that if these were equivalent in how much MD changed after stroke, that would argue that there's something about the cytoplasm, something about the viscosity of the water there that's different. However, if one was different than the other, in this case, if the longer diffusion time, if the change was greater there, that would argue for a micron-sized barriers or support the beating hypothesis. And what they found is that, in fact, it was the latter, that the pulse gradient spin echo was more sensitive to the changes than the oscillating gradient spin echo. And so what we did is performed again, these same simulations, but across a wide sampling of axon diameters of beating, and as well as intracellular volume fractions, and compared that data to identify where the human data best fit the simulation data. And so the answer is mean diffusivity. Is the decrease caused by beating or swelling? Yes. Unfortunately, it's likely to be both. But both were necessary to accurately mimic the DDLEI measurements using the simulations. This is another example of this. We're using a model of, in this case, they're taking diffusion kurtosis imaging, which I won't really discuss, but they're using a model to enable to resolve the intra-axonal diffusion coefficient from that of the extra-axonal diffusion coefficient, as well as a measure of the axonal water fraction here. And uniquely, what they showed here is a dramatic decrease in the intra-axonal diffusion coefficient, which would support the beating hypothesis, but they also saw a decrease in the extra-axonal diffusion. So again, it supports that there's probably at least two mechanisms at play here. So to revise this model of diffusion, I think it's important to point out that yes, if we have myelin damage, or yes, if we have axonal damage, this can lead to our corresponding increase in radial diffusivity or decrease in axial diffusivity. But the corollary is not necessarily true, right? So if we see an increase in radial diffusivity, that doesn't mean we're necessarily looking at demyelination. We have many other factors, including edema, CSF contamination, or inflammation that can contribute to this. So that's another point I want to make, is that if all we're looking at is the diffusion signal or the diffusion results, that doesn't implicate any one pathology over any others. We have to take some contextual information from the injury itself or the model that we're studying to really understand what's really going on, or try and formulate a reasonable hypotheses about what is happening microscopically. Again, axonal damage a decrease in axial diffusivity doesn't necessarily mean we have axonal damage. It can be inflammation or even partial volume effects, as we've seen earlier. So to wrap up, summary, fractional anisotropy, I think, as we've seen, it's not truly a marker of white matter integrity. And axial and radial diffusivity, while they provide some insight into DTI findings, as we've seen, DTI is really only good in a single coherent fiber population. But in my preference is for biologically inspired models to help us really get at some of these particular elements. However, again, we must understand that every model has their own assumptions, and we must be keenly aware of those when we're trying to interpret it. And lastly, I think I should point out, particularly in severe injuries, that tracks we're looking at are statistical connections and not necessarily continuous axons. So that needs to be reiterated. So with that, I want to acknowledge those sources and look forward to seeing you on the beach.
Thank you, Matthew. So do we have any question, still any doubt about the meaning of different diffusion contrasts now? Or are we now convinced about the histological validation? See, everyone is tired, actually looking to go to the beach probably now. So that was the last talk. And actually, I was thinking to invite all the speakers here. So just to say thank you all. So you want to come here? I'm just improvising. I have no idea what I'm doing, so let's see. Yeah. So actually, now that everyone is here, that was a trap, actually. And because all the experts are here, and my question is for you, if you have any doubt, any curiosity, we probably didn't cover today during the talk, now is the moment. So please, ask something, and we are here for you. I'm sure there is something you want to ask. Yeah, someone, are you asking when you can go to the beach? Well, I will say just after someone asks a question. Okay? So someone has to sacrifice. Come on. Well done.
Can I ask a question that was asked earlier, but there wasn't really a known answer yet? Maybe someone can make up their best answer.
Sure.
When you're screening for motion and all different pre-processing things, it's good at least to try to get through the review process to all kind of agree on certain types of practices. And so when you do identify bad gradients that you want to remove, it would be nice to have some type of rule of thumb that if more than 4% of your gradients are bad, toss the whole subject. If less than 4% are bad, just toss the gradients, and then we could kind of all follow that.
So it's a good idea. It's kind of difficult because it probably also depends on which model you're using, and
Yeah.
So coming up with a set of guidelines there is quite difficult because I mean, as you've noticed during this session, models are literally all over the place. There are hundreds of models nowadays. So, for, let's say, good old DTI, it might be more convenient to set up some protocol to do this.
Yeah.
But I'm not aware of any papers out there that, provide this information. I had a similar question indeed this morning. And again, if you've got poor quality gradient volumes, then with the robust estimators, ideally this information will be discarded anyhow. So that was my pragmatic, approach to tackle that. So.
Okay. Thank you.
If I can add something also, you also need to take into account all your data set, because if you have one or two bad subjects, probably be easier to just discard them. But then if you have 20 to 30% of your subject with a lot of artifact now, you might want to rethink to save as much as possible, so many as possible. So I think also depend on the data every time.
If you study a highly movable group of people, if you study a really high moving group of people like ADH children or elderly adults or something, you probably want to err on the side of removing gradients and not subjects.
Oh, sure. Well, I mean, that's the thing is, but the point is, as soon as you remove the gradient now, what you do when this, so you keep some subject with 30, 35 direction and some subject with only 20. So in terms of statistic, stability, you're going to have some effect, I think. So that's why you want to kind of balancing depending on how is your data. Yeah.
So can I if you have these kids that are moving a lot, then you can design your scheme file so that you can actually in your acquisition you can afford to lose some of the directions. And I think that instead of like when you're arranging the number of directions, I think it's Phil Cook, he has a paper on that, how you can organize your scheme file so if there's a lot of motion, you can just restrict and then you can still have a nice tensor fit. So that is one thing to account in that situation.
Yeah. Thanks.
One more thing too. So one thing that definitely is important is if the two groups or multiple groups are comparing, some of them actually more susceptible to motion, I think that will have a statistical effect, and I think that needs to be understood quite carefully. There's a recent paper, I can't quite remember the authors, it's in neuroimaging, about in children, in many of the studies of the children, where when you don't account for motion correctly, you actually find these consistent changes across many different disorders. But once you actually account for that, many of these differences disappear. And I think that do need to be taken, you know, in a serious way.
Yeah.
Okay, I think there is another question. Yep.
Yeah, before going to the beach, but supposedly a quick one. Just a question related to how should we work on validating the DTI results, and that seems to be there's a variability in terms of the approach that people are taking, post-mortem imaging or animal imaging, in order to validate all the measures and models. Is there any suggestion on how to move from one method to another? That means from post-mortem correlation to animal studies. What would be the main goal when you compare those two types of validation, if you know what I mean? So getting post-mortem data to validate or animal models to validate, they are very different from each other. So how would one go from one type of validation to another one, or even, is there any alternative solution for that? Because we have lots of information, but we don't know whether they are valid. I mean, we have different sources.
I can answer that. I think it ultimately depends on the question that you're asking. And the reason you're acquiring the data in the first place. So if you have some finding in the human and you want to say, okay, well, we see these changes, what are they? Then you take that to the animal model. Hopefully there's a good animal model that replicates the human situation. But it really depends on, yeah, I mean, really what you're going after. If you want to know what the pathology is, then you take that to the histology and try and do some work there. But if your question is very different, say you just want to say, well, is this relevant? Do I see a difference between two groups? I mean, maybe your DTI may be enough for that. So I think there's no one answer here, but it really depends on what you want to go after.
Yeah, for instance, FA models. It's one thing when you look at animal correspondence with FA, with many different animal models, not disease. I'm just saying that the source for DTI, I'm not talking about disease. Okay. But when you go to humans, again, normal tissue, you see again different results, because we all know that we have different methods. So I think as a field we should work on getting something that at least help us to make sense, because there's no other way to move around. As you just presented in many talks, there are many correspondences with the measurements, but it's really hard to make sense when you're talking about normal tissue. I'm not talking about disease so far.
I think that also speaks to repeatability amongst different groups. I mean, that's one thing that others have discussed here, but for example, if you really want to know the answer, having someone do a meta-analysis or things like that, I mean, a lot of different results are reported, but is what's being reported in your group the same as what's being reported in other groups? And that sort of speaks to the repeatability and reliability of those results. I mean,
so wouldn't be a time to make an effort to get something similar across groups with all these machines and you know?
Yes.
Sorry.
They are called multi-center study, and there are so many problems with that.
Yeah, so that was not a short question. Sorry about it.
Okay, thank you. Is there any other question? Yeah.
In some neurological diseases, subcortical nuclei, for example, such as thalamus, epithelium, and hippocampus is a major topic in measuring FA, but there are some methodological problems. Reduce the volume and preserve the axonal integrity, and ion concentration increase FA. So would you recommend any methodological caution in separating the FA value in subcortical nuclei and gray matter?
Sorry, can you just repeat the last part of the question? You're asking if what
gray matter
was about white matter integrity and its interpretation with FA.
Interpretation in subcortical nuclei, gray matter. Is there any caution, methodological caution?
So how we interpret FA in subcortical gray matter, and we can call it integrity or not, right?
Yeah.
So I think the question essentially, how can we interpret FA value not in white matter? So what is the meaning of FA value not in white matter? Is I mean, I think there is structural organization almost also in gray matter. We are actually, if we go in high resolution, high field, we also see clearly anisotropy in the cortex, and probably there inside the gray matter nuclei, we might see some structural organization. So I think the meaning of FA is not just white matter integrity, because we just saw that it's not that, the meaning of FA. Just FA is telling us only how much is the tensor anisotropic, how the model of diffusion is anisotropic. So it's about how you interpret this the meaning of your result according to your study. This is my answer. Don't know if anyone wants. You want to add something?
I think it's been pointed out that calling it white matter integrity is really kind of a misnomer. I mean, as we've seen, many different things can affect FA. If the fibers are more dense or there's injury. So I think, yeah, it really depends on, again, what your question is. And there's no way to interpret FA without some other context or without some other information.
Okay, if there are no more questions, I will say, again, thank you very much to all the speakers. It was a great day today. And, okay, now you can go to the beach, but before that, please, if you can go online and fill the evaluation form, because for us it's very important to see if we have done well or bad and how we can improve. So give us a lot of suggestions for next year, okay? Thanks again, thank you, bye.